Okay, so I want you to picture this. Imagine the sun has just slipped below the horizon. The warm tropical waters of the Indo-Pacific Ocean are going through this massive, almost theatrical transformation.
Right, because the daytime reef is so loud and chaotic.
Exactly. The bright, bustling coral reef of the day, with all its frantic energy, its darting clownfish and busy cleaner wrasses, it's all fading into these deep, shadowy shapes. The moonlight is just starting to filter down through the surface waves, casting this flickering silver glow over the coral rubble.
It gets so quiet.
So quiet. The daytime fish have tucked themselves away into rocky crevices to sleep. Some parrotfish have even spun little sleeping bags out of their own mucus to hide their scent. It's totally still. The frantic pace of the day is completely over. And down on the sandy bottom, you are just waking up.
You're just gliding over the sand. And I mean, to say you're walking wouldn't really be accurate. You are propelled by this single muscular foot just sliding over the uneven terrain.
Yeah, you don't have good eyesight, right?
No, not at all. Everything in your field of vision is just a blurry sense of light and dark, maybe a passing shadow. But out here in the pitch black of the ocean floor, you don't even need to see. You have a sensory tool that is, well, it's much, much better.
You extend this fleshy, striped tube out from your body into the gentle ocean current. It waves around slowly, side to side, almost like a magic wand, but it's actually acting as a super sensitive chemical radar. You are literally tasting the water.
Right, you're analyzing these microscopic molecules drifting in the current. And then suddenly you catch it. You smell the exact location of a small blenny fish.
It's fast asleep, hiding in a tight crack between two dead corals just a few inches away.
Now, you have to consider your physical limitations here. You happen to move slower than almost anything else on the entire reef. I mean, you are moving at an agonizing crawl. A sleeping fish could wake up and vanish in a fraction of a second.
But you aren't worried, are you?
Nope. You don't need speed, you just need chemistry. You creep closer, inch by silent inch, and you release a completely invisible, completely silent cloud of complex chemicals into the water.
And the sleeping fish breathes that invisible fog right in through its gills. Instantly, the fish gets incredibly dizzy. This heavy, unnatural sleepiness just washes over its entire nervous system.
It completely forgets how to move.
Yeah, its fins droop. It is totally frozen in this forced dream state. And that is when you make your move. You stretch your mouth out wider and wider until it looks less like a mouth and more like a massive ghostly net. You sweep it entirely over the sleeping fish, swallowing it whole right there in the dark.
It's a perfectly executed, completely silent ambush. And it's orchestrated by an animal that lacks a brain, lacks a spine, and lacks the ability to move faster than a few inches per minute.
Welcome back to Saltwater Stories. Today we are exploring the secret life of the ocean's ultimate nighttime ninja. You just experienced a hunt from the perspective of Conus geographus, the geography cone snail.
It is such an incredible creature.
It really is. It moves at a literal snail's pace, yet it stands one of the most successful and honestly most dangerous predators in the entire ocean. And we are so, so glad you're here with us to uncover its secrets.
You know, the contrast between our expectation of a predator and the reality of this snail is just astounding.
Oh, totally. I mean, when we think of apex predators, we immediately jump to like great white sharks with rows of serrated teeth, right?
Exactly. Or saltwater crocodiles with thousands of pounds of bite force. We expect immense size, terrifying speed, and brute strength. But this snail proves that sometimes the most spectacular, lethal survival skills belong to the slowest, most unassuming creatures in the sea.
I love that. Let's hold on to that thought about spectacular survival skills, because before we even get into the invisibility cloaks and the sleeping potions we just talked about, I want to share another quick story about this animal.
Oh, the weapons factory.
Yes. I want you to imagine a creature that has a literal microscopic weapons factory built right inside its own body. And this factory operates continuously, building these microscopic hollow swords.
The manufacturing process inside this snail is just a marvel of biology. It doesn't just grow a sharp tooth.
Right, it's way weirder than that.
It starts by growing flat, tough plates. Then, inside a specialized sac, it rolls those flat plates up, almost exactly the way you would roll up a heavy living room carpet, until they form a perfectly hollow, structurally rigid tube.
It rolls up these hollow swords, stores them in an internal quiver, and when it is time for dinner, it loads one of these microscopic swords into a flexible trunk. It fires the sword like a high-speed harpoon to catch its meal.
And here's the kicker. It uses that specific, custom-built sword exactly once.
Mind-blowing. When it's done eating, it just casually spits the used disposable weapon out into the sand and loads up a fresh one for the next night.
The level of biological engineering required to maintain a disposable weaponry system is staggering. Today, our goal is to really unpack the mechanics of how this incredible animal pulls off these supervillain-level gadgets.
Yeah, we're going to examine why it is so vitally important to the delicate balance of the ocean and how its deadly chemical weapons might actually hold the blueprint for human medical miracles.
It's going to be a fascinating journey.
I am thrilled to get into this. Let's start with the visual. What does this creature actually look like? Like if you were walking down a beautiful tropical beach at low tide and you saw the bare shell of a geography cone snail washed up on the sand, your first instinct would probably be to put it right in your pocket.
Oh, absolutely. It's stunning.
It is. Imagine taking a delicate porcelain teacup and drawing an intricate ancient pirate map all over it.
Yeah.
That is what the shell looks like. It features these beautiful chestnut and white patterns, forming little swirling islands and continents and topographical lines.
And that specific map-like pattern is actually why the famous scientist Carl Linnaeus named it Geographus way back in 1758.
Geographus like geography.
Oh, yeah.
Makes perfect sense.
But there is a massive catch to that historical naming. If you were swimming in the ocean, actively looking for a living geography cone snail, you would almost certainly never see those beautiful map patterns.
Wait, why not?
Because Carl Linnaeus named the species based on a clean, empty, polished shell that an explorer brought back to a pristine laboratory. Out in the wild, the living snail wears something akin to an invisibility cloak.
An invisibility cloak? That sounds like magic, but I'm guessing this is an evolutionary adaptation, right? How does a snail make itself invisible?
It creates an outer layer that completely covers the shell, which scientists call the periostracum.
Periostracum. Okay.
It is a thin, yellowish, highly textured skin made entirely of protein. But it doesn't lay flat against the shell. It forms these little hair-like tufts and ridges all over the exterior, which gives the snail a very rough, mossy, almost fuzzy appearance.
Oh, if it's not smooth at all.
Not at all. And because it has this rough protein skin, tiny marine plants like algae and microscopic organisms can latch onto it and grow right on the snail's back.
So it is actively cultivating a garden on its own back. It's wearing a biological ghillie suit.
Exactly.
It takes this beautiful matte-painted shell and disguises it to look completely identical to a boring, algae-covered rock sitting on the bottom of the ocean.
The camouflage is flawless. And it is a necessary defense mechanism because of the specific environment it inhabits. You will find these snails scattered throughout the tropical Indo-Pacific.
That's a huge area, right?
Massive. It's a warm water expanse, stretching from the eastern coast of Africa, across the Indian Ocean, all the way to the northern coast of Australia, and out into the remote Pacific Islands.
And they especially hang out in a region called the Coral Triangle. Marine biologists sometimes refer to the Coral Triangle as the Amazon of the seas.
It's a great nickname for it.
Yeah, because it is packed with just an incomprehensible amount of biodiversity. We are talking about thousands of competing species of fish, intricate coral structures, and invertebrates, all fighting for survival in the exact same space.
And within that massive triangle, they specifically live in what scientists call the sublittoral epipelagic zone.
Okay, let's translate that terminology for a second. Sublittoral epipelagic zone. Does that basically just mean the shallow, sunlit waters near the beach? Like the areas where the coral reefs are close enough to the surface to soak up the sun?
That is a perfect translation, yes. The water there is warm, it is shallow enough for sunlight to penetrate all the way to the bottom to feed the corals, and the seafloor is a complex mixture of coral rubble, rocky outcroppings, and sandy patches.
But wait, I'm finding a contradiction here.
Oh, what's that?
Well, if the snail lives in this beautiful sunlit coral reef, and it has evolved this incredible invisibility cloak that makes it look exactly like a rock, why does it hide all day?
I see what you mean.
Like during the daylight hours, it completely buries itself under dead coral or sand and only emerges to hunt during the night shift. If its camouflage is so perfect that no one can see it, why not just hunt during the day?
That is an excellent question. And it comes down to the behavior of its prey and the snail's own metabolism. Even with world-class camouflage, the daytime reef is a chaotic, high-stakes environment.
Lots of predators roaming around.
Exactly. There are large visual predators like crabs, octopuses, and certain fish that might accidentally stumble upon the snail or detect its movement. But the primary reason is the fish it hunts. A daytime reef fish is fully alert, visually sharp, and incredibly fast.
Oh, right. A snail, no matter how invisible, cannot sneak up on a fish that is awake and darting around.
It absolutely requires the cover of darkness to find fish that have powered down to sleep.
So it's a matter of timing. It has to wait for the target to be totally immobile.
Yes. Additionally, there is a fundamental biological constraint. Cone snails are heterothermic.
That means cold-blooded, right?
Exactly.
Yeah.
It's a specialized term, meaning their body temperature entirely dictates and is dictated by the temperature of the ocean water around them. They cannot generate their own internal body heat like a mammal.
Right, like how we sweat or shiver to stay the same temperature. They just match the water.
Correct. By resting buried in the cool sand during the heat of the day and only exerting energy at night, they keep their metabolic rate perfectly balanced with their surroundings, conserving massive amounts of energy.
Okay, so we have a cold-blooded, rock-disguised ninja that only comes out when the sun goes down. But to truly understand how a creature that moves this slowly can hunt vertebrate fish that move like lightning, we really have to look inside its shell.
The internal anatomy is where things get truly bizarre.
Yeah, because its body operates on completely different principles than our own bodies. It's built like an alien spacecraft.
The internal architecture of a gastropod, which is the overarching class of mollusks that snails belong to, is wonderfully alien compared to mammalian biology. Let's examine the engine that drives this creature, its circulatory system.
Okay, humans and the fish the snail hunts have a closed circulatory system.
Right. Our blood flows neatly and directionally inside a vast network of closed tubes. Our veins, arteries, and capillaries, all pumped by a powerful, four-chambered heart.
A closed system is very organized. It's high pressure, kind of like municipal plumbing pushing water to every house in a city.
But the geography cone snail utilizes an open circulatory system. It doesn't pump red, iron-rich blood like we do. It utilizes a fluid called hemolymph.
Hemolymph, basically snail blood.
Exactly. And instead of being restricted to miles of tiny veins, this hemolymph is pumped by a very simple, low-pressure, two-chambered heart. The heart pumps the hemolymph out, and it simply washes entirely over the snail's internal organs.
Wait, it just washes over them?
Yes, it pools in large body cavities before eventually making its way back to the heart.
Let me make sure I'm picturing this correctly. Instead of blood traveling through pipes to reach the stomach or the liver, the snail's organs are essentially just soaking in a warm, continuously circulating bath of snail blood.
That is a highly accurate way to visualize it.
That is so weird.
It is. But for an animal that doesn't need to run, jump, or sustain prolonged aerobic exercise, a low-pressure open circulatory system is incredibly energy efficient. But the circulatory system is only half of the alien anatomy.
What's the other half?
The truly mind-bending part is how the snail processes information and reacts to its environment.
Oh, the nervous system. This is the part that fascinates me. Because think about how human beings are built. We have a brain centralized in our head. It is our ultimate command station.
Right. Everything routes through the brain.
Yeah, every time you touch something hot, a signal has to travel all the way up your arm, into your spinal cord, up to your brain, get processed, and then a signal has to travel all the way back down to your hand to tell your muscles to pull away.
Which takes a fraction of a second, but it is a long journey biologically.
But if you asked an engineer to build a hyper-efficient, reflex-driven robot, they probably wouldn't put all the processing power in one single location. They would spread mini computers all over the robot's body so different parts could react instantly without waiting for a central command.
And that seems to be exactly what the snail does. The robotics analogy highlights the efficiency perfectly. The geography cone snail completely lacks a traditional centralized brain.
No brain at all, just wild.
Instead, its nervous system is decentralized. It relies on a nerve ring made up of distinct structures called ganglia.
Ganglia. Let's break that down for our listeners. Are they basically just knots of nerves?
You can think of them as specialized processing hubs. These ganglia are clustered around the snail's esophagus or throat area, and different knots are dedicated to controlling entirely different physiological functions.
So they divide up the work.
Precisely. One ganglion strictly controls the movement of the muscular foot, another oversees the sensory organs, and a completely separate one manages the venom delivery system.
By having these mini control centers separated but networked, the snail can execute lightning-fast reflex reactions.
Exactly. The foot doesn't need to consult a central brain to know how to move based on a sensory input. The local ganglion handles it instantly.
Which explains how it can trigger a complex hunting mechanism so rapidly. But before it can fire any weapons or move its foot, it has to actually locate the fish.
That is the first crucial step.
And we established earlier that its eyesight is practically non-existent. It just has these two tiny, primitive stalk eyes that do little more than differentiate between light and shadow. So how is it finding a tiny fish hiding in a massive, dark coral reef?
It relies heavily on a specialized organ called the osphradium.
The ospradium. I have to admit, that sounds like a rare metal you'd find on the periodic table, not a body part. What does an ospradium actually do?
It functions as an underwater supernose, but it operates on a chemical level that is hard for us to fathom. Recall the fleshy, striped tube waving in the water from our opening story, the siphon.
Right, the magic wand.
Exactly. The snail actively draws ocean water into that siphon, and that water is channeled directly over the osphradium. This organ is incredibly densely packed with chemical receptors.
So it's literally tasting the water.
Yes. It is highly sensitive to the microscopic amino acids and metabolites floating in the water that are constantly shedding off the body of a resting fish.
So it's actively pulling water in, running it across this sensory organ, and sniffing out the invisible chemical trail of a sleeping blenny fish.
And it follows that scent gradient, moving toward the highest concentration of amino acids right to the crevice.
But here's the problem I'm seeing. The fish might be asleep, but it is still a fish. If it wakes up, it can accelerate and be gone in a split second.
True.
The snail physically cannot chase it. So the supernose has found the target. How does the snail secure the catch without triggering the fish's flight response?
It secures the catch by fundamentally altering the chemistry of the surrounding water, unleashing a chemical fog that forces the fish into a metabolic dream state.
The Nirvana Cabal. I absolutely love this biological term. It sounds like a secret society of wizards in a fantasy novel, but it's a real scientific phenomenon. Let's explore how this works.
It is a staggering piece of evolutionary biochemistry. When the snail is in close proximity to the sleeping fish, it slowly releases a highly complex mixture of hundreds of tiny, powerful proteins into the water.
And these proteins are called conotoxins, right?
Yes, conotoxins. This specific preliminary mixture, the one released into the water as a fog rather than injected, is formally called the Nirvana Cabal. The word nirvana refers to a state of blissful, detached sleep or transcendence, and cabal implies a secret group of agents working closely together to achieve a specific outcome.
And the most mind-blowing part of this secret group of toxins is the primary ingredient. It is weaponized insulin.
Yes.
Now, wait a minute. Let's pause and look at this logically. Many people are familiar with insulin. Humans use insulin to manage diabetes. It is a crucial hormone that regulates blood sugar so our bodies have energy.
Okay.
How on earth does a snail use a blood sugar hormone as an offensive biological weapon? Wouldn't giving a fish a dose of insulin just give it a massive energy boost? I mean, I'm having trouble seeing how this puts a fish to sleep.
It's a great question. To understand the weaponization, we have to look closely at what insulin actually does at the cellular level. Whether you are a human or a reef fish, your body requires energy to function, and that energy primarily comes from glucose sugar circulating in your bloodstream.
Right, sugar is fuel.
But that sugar cannot simply phase through the walls of your muscle cells. The cells essentially have locked doors. Insulin is the biological key that fits into the lock, opening the cell doors and allowing the sugar to leave the bloodstream and enter the tissues to be burned for energy.
Okay, the lock and key mechanism makes sense. Insulin opens the door. But how does opening the door hurt the fish?
Well, the geography cone snail produces a highly specialized lab-grade venom insulin. Scientists have designated it Conins G1.
Catchy name.
Right. This venom insulin has been structurally adapted over millions of years to look absolutely identical to the fish's natural insulin. It is a perfect counterfeit key. The snail releases a massive, overwhelming dose of this counterfeit insulin into the water. As the sleeping fish breathes, drawing water over its highly vascular gills, the venom insulin absorbs directly into the fish's bloodstream.
Ah, I see the mechanism now. Because the fish's body is suddenly flooded with all these counterfeit keys, it thinks, oh, we have a massive amount of insulin, time to open every single door in the body.
Exactly.
And all of the sugar in the fish's blood gets pulled out of the bloodstream instantly.
Precisely the opposite of an energy boost. By forcing all the sugar out of the blood at once, the fish's blood sugar plummets to near zero in a matter of seconds.
Wow.
This induces a severe, acute medical crisis known as hypoglycemic shock. The brain and the central nervous system rely entirely on a steady supply of blood sugar to function. Without it, they starve.
So the fish just crashes.
The fish becomes incredibly lethargic. Its cognitive functions shut down and becomes deeply sedated, and it completely loses the physical ability to coordinate its muscles to swim or escape.
That is diabolical. But what really blows my mind is the specific molecular engineering the snail uses to make this happen so fast. Because human insulin and the natural insulin found in normal fish does not work instantly. If a person takes insulin, it takes time to take effect.
Yes, the delay in natural insulin is due to how the body stores it. Natural insulin is produced and stored in the pancreas. To pack it tightly and store it safely without it degrading, the body clumps the insulin molecules together in highly organized groups of 6, which biologists call hexamers.
Like taking 6 sticky Lego bricks and snapping them all tightly together so they fit neatly into a compact storage box.
The Lego analogy is quite fitting. But when your body actually needs to utilize that stored insulin to manage a meal, it takes significant time to unsnap those complex Lego structures.
Because they're all stuck together.
Right. The hexamers have to be chemically broken down from groups of 6 into groups of 2, and finally into single unattached molecules monomers before they can actually fit into the cellular locks. That unsnapping process can take up to an hour.
But the snail doesn't have an hour. If it releases the fog and has to wait an hour for the insulin to break down, the sun will come up, the fish will wake up, or another predator will come along and ruin the ambush.
And evolution solved the storage problem. The snail's venom insulin is genetically engineered to be fundamentally missing the specific molecular peptide, the sticky edge that causes the insulin to clump together into hexamers.
So it doesn't stick together at all.
Nope. It is synthesized and stored entirely as single, unattached molecules.
So the snail bypasses the unsnapping process entirely. It is firing single, ready-to-use keys right into the fish's gills.
Because it doesn't have to break down from a hexamer state, the snail's venom insulin hits the fish's system and begins unlocking cells in under 5 minutes. The sheer speed of the chemical reaction is what makes it a viable weapon.
That is just, it's beyond science fiction. And as if deliberately crashing the fish's blood sugar to 0 wasn't a thorough enough strategy, the Nirvana Cabal actually has a backup plan built in, doesn't it?
It does.
There is another chemical involved called consumatin NG1.
The redundancy in the venom is terrifyingly effective. In a normal scenario, if a fish experiences a sudden, dangerous drop in blood sugar, its body has an automatic emergency alarm system.
Like a backup generator.
Exactly. The pancreas releases a different hormone called glucagon, which signals a liver to immediately dump emergency sugar reserves back into the bloodstream to save the animal's life.
A biological failsafe.
Consumatin NG1 is a molecule specifically designed to disable that failsafe. It binds to the receptors and explicitly blocks the emergency alarm.
Cuts the alarm wire.
It completely stops the fish's body from releasing the rescue sugar. It ensures the hypoglycemic shock is sustained, and the fish absolutely cannot wake itself up.
So let's look at the situation for the fish at this exact moment. The fish is floating in a total daze. It has absolutely no usable energy in its brain. Its emergency rescue alarms have been severed, and it is practically in a coma with its eyes open.
The trap is sprung.
It is time for the snail to cast its net. Let's talk about the physical mechanism of swallowing the fish.
Yeah.
The false mouth.
The anatomical term for the false mouth is the rostrum. And to fully appreciate how the geography cone snail utilizes its rostrum, we should contrast its hunting style with how the majority of other cone snails operate.
Because there are hundreds of different species of cone snails, right? And many of them use a much more aggressive strategy that marine biologists call the taser and tether method.
The taser and tether method is highly dramatic. Picture a different species of cone snail extending a long muscular tube, violently firing a harpoon into a fish that is actively swimming by.
Ouch!
And then using a strong filament attached to the harpoon to physically reel the thrashing, fighting fish into its mouth, much like an angler fighting a marlin on a fishing rod.
But our snail, Canis geographus, doesn't fight its prey. It uses net hunting.
Because the prey is already profoundly sedated by the Nirvana Cabal, the snail doesn't need to shoot it from a distance or fight to reel it in. Instead, it slowly pushes its rostrum out.
And the rostrum is incredibly elastic and stretchable. It expands outward like a massive, fleshy, translucent balloon.
It really does.
It's an incredible visual. It's almost like a ghost stretching its mouth to impossible proportions to swallow a car. The rostrum just expands and expands until it completely wraps around the sleepy fish.
And because the fish is so heavily sedated and unaware, sometimes the geography cone snail can swallow an entire school of small resting fish at the exact same time, trapping all of them inside this dark, fleshy net.
Oh, wow, a whole school at once. But The prey is completely engulfed, but they aren't dead yet, right?
Correct. They are merely incapacitated.
And containing a live wild fish, even a sleepy one, inside your digestive tract seems like an incredibly dangerous position for a soft-bodied invertebrate.
Reef fish have highly specialized defenses. They have razor-sharp dorsal fins, spiky scales, and bony gill plates.
Exactly. If the fish were to suddenly wake up or thrash reflexively inside the snail, those spines could easily slice the snail's soft stomach open from the inside out, which would be fatal. The snail has swallowed a bomb that could go off. Which brings us back to the second story we started with, the disposable sword factory. It is time for the snail to build and fire the harpoon.
Let's look at how the weapon is constructed. Most common garden snails have a structure called a radula. It's essentially a rough, muscular tongue covered in microscopic hard bumps, which they use to scrape algae off of rocks and leaves.
Like a little white piece of sandpaper constantly scraping away.
But the geography cone snail's radula has undergone a radical evolutionary divergence. Inside a specialized pouch called the radular sac, the snail continuously grows teeth.
And these teeth start out flat, right?
Yes, they start out as flat, two-dimensional plates made of chitin and calcium. As they mature and move up the assembly line, the plates fold and roll up onto themselves, creating incredibly sharp, perfectly hollow hypodermic needles.
Microscopic needles made of rolled-up teeth. That is wild.
When the fish is safely enveloped inside the expanded rostrum, the snail moves one of these hollow harpoons into its proboscis. The proboscis is a highly flexible, muscular firing tube located deep within the mouth cavity.
I'm picturing a biological turkey baster. At the back of this whole venom delivery system, there is a thick, highly muscular venom bulb filled with a completely different toxic fluid.
Yes.
When the snail is ready, it squeezes that venom bulb with immense force. That intense pressure forces the venom down the tube, pushing the hollow harpoon out with incredible velocity.
The physics of the strike are astonishing. High-speed macro photography has revealed that the harpoon fires out of the proboscis in less than 200 microseconds.
Let's put 200 microseconds into context. That is a fraction of a millisecond. That is comparable to the speed of a bullet leaving the chamber of a firearm.
It is remarkably fast.
It is one of the absolute fastest biological movements recorded in the entire animal kingdom, and it is happening deep inside the false mouth of a creature that moves an inch a minute.
The hollow harpoon violently pierces the sedated fish, and the hydraulic pressure instantly injects a secondary, entirely different venom mixture. This secondary venom is known as the motor cabal.
So the first venom was the nirvana cabal, designed to crash the blood sugar and induce sleep. The motor cabal is designed to do exactly what it sounds like, attack the motor functions.
The chemistry of the motor cabal is brutal. It is packed with excitotoxins that specifically target the voltage-gated sodium channels in the fish's nervous system.
What does that mean for the fish?
Well, when these channels are jammed open, the fish's muscles receive a continuous, unbreakable signal to contract. It instantly freezes the fish stiff. This is known medically as tetanic paralysis.
Wow.
Every muscle in the fish's body locks up simultaneously, making the fish perfectly rigid.
So it transforms from being a sleepy, dangerously floppy fish into a frozen fish stick.
Essentially, yes.
And that is exactly why the snail waits until the fish is completely engulfed inside its mouth before stinging it. It freezes the fish solid so those sharp fins and spines are locked in place and cannot thrash around to cut the snail's soft internal organs.
It is a meticulously calculated sequence of events. Sniff out the chemical trail, drug the water to crash the metabolism, cast the fleshy net, swallow the prey whole, fire the microscopic bullet, freeze the prey solid. The hunt is over.
Just incredible. And there is a final fascinating piece of biological housekeeping to this process, isn't there?
There is. The snail's digestive enzymes quickly break down the soft tissues of the fish, but it cannot digest the hard keratinous scales, the calcium-rich fish bones, or its own chitinous hollow tooth.
So what does it do with all that trash?
Several hours, or sometimes even days after the meal, the snail physically regurgitates. It throws up a neat, compacted little package containing all the undigested bones, scales, and the spent harpoon, casually spitting it out onto the sand.
The ultimate disposable weapon. Use it once, throw the casing away, and load up the next one for tomorrow night.
Exactly.
But, stepping back from the hunt, a realization hits. You don't just wake up one morning with a complex internal weapons factory, a protein-based invisibility cloak, and lab-grade weaponized insulin.
No, you certainly don't.
You don't achieve this level of predatory perfection by practicing for a few weeks. You practice for 55 million years. Let's delve into how these snails grow up and the evolutionary journey that forged them.
The life cycle of the geography cone snail is a sweeping narrative of extreme vulnerability, high mortality, and dramatic biological transformation.
Right, because adult snails are geographically restricted by their incredibly slow pace. They cannot simply migrate to a new coral reef to find a mate or establish a new territory.
They must rely on the ocean itself to disperse their offspring across the globe.
So how does the process start?
It starts with the mother snail.
Hmm.
She locates a relatively safe, secluded spot, perhaps tucked deep under a heavy piece of coral rubble, and she secretes and glues down dozens of tough, leathery egg capsules.
And she can deposit thousands of eggs in a single reproductive event, right?
Yes. And then she departs. There's absolutely no parental care or babysitting in the cone snail world.
Harsh. So what happens to the eggs?
The embryos develop inside these protective capsules for a period of a few weeks. However, when they finally hatch, they do not emerge as miniature, crawling snails with little shells.
No.
They hatch into a completely different morphological state. They emerge as microscopic, transparent, free-swimming creatures known as villager larvae.
Villager larvae. So instead of crawling on the sand, they're just these tiny translucent specks floating aimlessly in the water column of the open ocean.
They possess tiny hair-like appendages called cilia, which they use to maintain their position in the water and to sweep microscopic planktonic food toward their primitive mouths.
But this brings up the massive logical question. We just spent all this time establishing that the adult geography cone snail is a terrifying, highly optimized apex predator. Why on earth would it start its life floating helplessly in the open ocean, completely exposed, where literally everything from massive filter-feeding baleen whales to hungry coral polyps to every single small fish can just gulp them down by the thousands? It seems like a catastrophic survival strategy.
It appears incredibly dangerous, and statistically, it is. The vast majority of those thousands of larvae will be consumed within days and will never reach adulthood.
Then why do it?
Biologically, it is a brilliantly calculated evolutionary trade-off. By floating in the pelagic zone, the open ocean water column, the larvae surrender themselves to the massive global ocean currents.
Oh.
These currents act as a superhighway, carrying the surviving larvae hundreds, sometimes thousands of miles away from their natal reef.
That perfectly explains their massive geographic distribution. That is how they spread all the way from the coast of Africa to the reefs of Australia. They surf the global currents as invisible babies.
The ocean currents guarantee the survival and expansion of the species. Now, for the incredibly lucky fraction of a percent of larvae that survive this harrowing drifting phase, they eventually grow heavy enough to settle out of the water column and sink down to the sandy bottom of a new coral reef.
And that's where the magic happens.
Here, they undergo one of the most remarkable changes in the animal kingdom.
The ontogenetic shift. This is a fascinating concept in biology.
An ontogenetic shift refers to a fundamental, dramatic change in an organism's diet, behavior, and physical morphology as it develops from a juvenile to an adult.
So when the baby snail finally settles onto the sand and begins calcifying its hard, patterned shell, it is minuscule. It is a fraction of an inch long.
At that size, attempting to catch or swallow a vertebrate fish is a physical impossibility.
Yeah, a fish would just look at it and swim away or, eat the snail itself.
Therefore, the juvenile geography cone snail does not hunt fish at all. It survives by hunting slow-moving, relatively defenseless marine worms.
It's like biological training wheels. It practices the mechanics of hunting on a target that can't fight back.
But as the snail matures and increases in size, its internal anatomy undergoes a complete rewiring. The shape of the teeth produced in the radular sac shift dramatically, growing from short, stout spikes suited for piercing soft worms into the long, sophisticated, hollow harpoons necessary for injecting fish.
It completely changes its own weapons.
Even more astonishingly, the cellular factories within the venom duct actually alter the chemical recipe of the venom itself. They transition from a simple worm-killing poison to the immensely complex, multi-tiered nirvana and motor cabals required to shut down a vertebrate nervous system.
The animal literally edits its own internal biology and chemical engineering as it grows to support a higher-tier diet. That is so cool.
It really is.
But how did the species acquire this ability over deep time? I mean, if we rewind the evolutionary clock 55 million years, the fossil record and genetic tracing suggest that the ancient, distant ancestors of all cone snails were simple bottom-dwelling worm hunters.
That's right.
How does a species make the monumental leap from eating a slow, squishy worm to chemically sedating and harpooning a hyper-fast, complex fish.
The transition is a textbook illustration of a profound evolutionary concept known as the Red Queen hypothesis.
The Red Queen hypothesis. This draws its name from Lewis Carroll's Through the Looking Glass, the sequel to Alice in Wonderland, right?
Yes, exactly.
Where the Red Queen tells Alice that in her kingdom, it takes all the running you can do just to stay in the exact same place.
The metaphor is incredibly apt. Visualize evolution not as a ladder, but as a treadmill. Millions of years ago, an ancestral cone snail might have been born with a random genetic mutation that made its venom just a tiny bit faster acting. Or perhaps it affected a slightly different nerve pathway.
Right, a happy accident.
That specific snail could suddenly incapacitate a slightly faster prey item, perhaps small, sluggish, bottom-dwelling fish. Access to this new calorie-dense food source gave that snail a massive survival advantage, allowing it to thrive and pass that mutated gene on to its offspring.
But the ecosystem doesn't just accept defeat. The fish don't volunteer to be eaten.
No, they don't.
The fish that were naturally a little faster or had nervous systems naturally a little more resistant to the primitive poison were the only ones that survived to reproduce and have fish babies.
Consequently, the next generation of fish is inherently faster and more resistant. To continue catching them, the snail must evolve an even faster, more chemically complex venom. The fish respond by evolving better biological defenses.
It is a continuous 55 million year-long biological arms race. The snail is forced to constantly innovate its chemical weapons just to maintain its position in the food web, running endlessly on the evolutionary treadmill.
And the specific molecular mechanism the snail used to innovate its weaponized insulin is fascinating. Biologists call the process gene duplication.
Gene duplication. Let's dig into that.
Gene duplication is a powerful driver of evolutionary novelty. Like all complex organisms, the snail absolutely requires its own natural functioning insulin hormone to manage its own cellular energy. It cannot survive without it.
Makes sense.
But deep in its evolutionary past, during the process of cell division, an error occurred. The snail's DNA accidentally made a second, identical copy of the insulin gene.
A biological typo. It hit copy and paste on its own DNA sequence.
The typo provided an incredible opportunity. The snail kept the original functional copy of the insulin gene safely integrated in its nerve ring to keep its own metabolism running smoothly.
So it didn't lose its natural insulin?
Exactly. But the duplicate extra copy was free from the pressure of keeping the snail alive. It was relocated to the venom gland, where it was subjected to millions of years of rapid, aggressive mutation.
Wow.
The snail essentially tinkered with this backup file, altering molecular structure generation after generation until the resulting protein perfectly mirrored the insulin of the fish it was trying to consume.
It utilized a stolen, mutated blueprint to pick the metabolic locks of its prey. It is sheer brilliance. But, we have to acknowledge the dark side of this evolutionary perfection. Because this venom has been honed over 55 million years, specifically to instantly bypass and shut down the complex vertebrate nervous systems of fish.
Yes, which leads to a dangerous cross-compatibility.
Right. It means the venom is inherently incredibly dangerous to any other organism with a similar vertebrate nervous system.
Which heavily implies mammals, including human beings.
Right. Let's transition to human encounters, because I want to be abundantly clear and make sure we emphasize this heavily to everyone listening. Geography cone snails do not hunt humans.
They absolutely do not.
They have absolutely no desire to eat us. We are vastly too large to fit inside a rostrum. They do not track us.
That is a critical distinction. If a human being is stung by a geography cone snail, it is purely an accidental threat. The fault lies entirely with the human. The snail is simply reacting defensively because a massive, unrecognizable force has disturbed it, and it is terrified for its life.
What I find so intriguing about this defensive reaction is that the snail actually possesses the cognitive ability, or at least the hardwired reflex, to differentiate between hunting for food and fighting for its life.
It is a remarkable display of biological resource management. We discussed the nirvana cabal used for sleeping prey and the motor cabal used for freezing them.
Right?
Recent chemical analysis has revealed that the snail actually produces two entirely distinct venom profiles stored in different areas of the duct, depending on the immediate situational context.
Wait, really? Two different venoms ready to go?
Yes. When it is executing a hunt, it utilizes the hunting venom, the weaponized insulin, and sedatives. This hunting venom is designed to be relatively painless. Its sole purpose is to make the prey fall asleep without alarming it.
But if a giant human hand plunges into the water, grabs the shell and rips the snail off the sandy ocean floor.
The snail's sensory ganglia immediately register a catastrophic threat. It instantly switches protocols, routing a different chemical mixture to the harpoon. It deploys its defensive venom.
And the defensive venom is different.
Completely different. This mixture is a highly concentrated cocktail of paralytic toxins and pain-inducing peptides. The defensive venom is biologically designed to cause immediate excruciating pain and rigid paralysis in order to shock and deter large, crushing predators like crabs, stingrays, or sea turtles.
And when a human nervous system gets hit with that defensive venom, it initiates an immediate, dire medical emergency.
It does.
Let's look at the historical record because it underscores the gravity of the situation. There is a well-documented, famous case from 1935 involving a 27-year-old man named Charles Garbutt. He was vacationing at Hayman Island, which is located right on the Great Barrier Reef in Australia.
A pristine, beautiful location, perfectly suited for exploring the diverse tide pools at low tide.
Charles was walking along the exposed reef, peering into the pools. He spotted this gorgeous, intricately patterned shell resting on the bottom. Assuming it was empty, or perhaps just wanting to admire the pattern, he reached down, picked it up, and began to scrape away the rough periostracum to clean the shell. He had no idea there was a live, highly venomous animal retracted deep inside. The snail, feeling the vibrations of being scraped and held, extended its proboscis out of the shell and fired a defensive harpoon directly into the palm of his hand.
And the medical progression of the symptoms following a defensive sting is terrifyingly rapid. Because the conotoxins are engineered to immediately bind to and shut down nerve channels, Charles experienced profound numbness in his lips and face almost immediately after the strike.
That fast?
Yes. Within an hour, his visual cortex was affected, causing his vision to fade, and the excitotoxins began locking up the sodium channels in his limbs, causing total rigid muscular paralysis.
And the fatal flaw in human anatomy when exposed to this venom is that the paralysis doesn't just affect the arms and legs, does it?
No, it doesn't.
The venom systematically paralyzes the diaphragm, the massive muscle located below the lungs that we involuntarily use to pull air into our bodies. Despite medical personnel arriving to assist, Charles physically could not expand his lungs to breathe, and he tragically passed away.
It's incredibly sad.
To this day, it remains Australia's only medically confirmed, documented fatality from a cone snail sting. But it serves as a sobering, powerful reminder of the chemical potency this animal wields.
And we must emphasize a terrifying medical reality regarding these encounters. There's absolutely no anti-venom available for a cone snail sting.
None at all. That's so scary. I mean, if a person is bitten by a highly venomous snake or a spider, emergency rooms can often administer a specific antivenom, a serum containing antibodies that physically bind to and neutralize the poison circulating in the blood. Why can't we do that for the snail?
The barrier is the sheer complexity of the venom. Snake venom typically consists of a handful of primary toxic proteins. The geography cone snail's venom is an ever-shifting, hyper-complex cocktail containing hundreds of distinct, highly specialized peptides.
Too many ingredients to fight.
Exactly. Attempting to create a single antivenom that can simultaneously neutralize hundreds of different, rapidly acting neurotoxins before they bind to the nervous system is currently beyond the capabilities of modern pharmacology.
So if there is no antivenom, what is the medical protocol? If someone is stung, how do doctors save them?
The treatment is entirely supportive. Because the primary cause of death is asphyxiation due to diaphragm paralysis, doctors must immediately intubate the patient and connect them to a mechanical ventilator.
So the machine breathes for them?
Yes, the machine physically forces oxygen into the patient's lungs, taking over the work of breathing. The medical team simply has to keep the patient oxygenated and alive for hours or sometimes days until the human body's liver and kidneys can naturally break down the complex conotoxins and flush them out of the system.
And once the venom degrades, the paralysis lifts and the patient can breathe independently again.
That's right.
Knowing all of this, how do we interact with the ocean safely? Because exploring tide pools, snorkeling over reefs, and discovering marine life is one of the greatest joys of interacting with nature. We want people to explore. We just need to adhere to the golden rule of marine biology.
The rule is simple and absolute. Look, but do not touch.
Let's establish the 3 no's of ocean safety for anyone exploring a reef. Number 1, no picking up unknown shells. Even if a shell looks completely clean and empty, marine snails have incredible muscular control and can retract their entire bodies way deep inside the spirals where you cannot see them.
Always assume it's occupied.
Exactly. Number 2, no touching creatures hiding in crevices. If you see something tucked in a rock, leave it be. And number 3, No reaching your bare hands into murky tide pools where you cannot clearly see the bottom.
The ocean is a dynamic, wild ecosystem, not a curated petting zoo. The creatures that inhabit it possess defense mechanisms honed over millions of years, and they command our utmost respect and distance.
Absolutely. But here is where the narrative of the geography cone snail takes the most unexpected magical twist of all. That exact same deadly defense venom, the precise chemical cocktail that causes rigid paralysis, the mixture that makes the snail so feared, is currently sitting in modern, sterile science laboratories, actively saving human lives.
It is the ultimate profound irony of the natural world. The ocean's deadliest, most refined chemical weapons are being reverse engineered into our greatest, most effective medicines.
I am fascinated by this concept of the ocean pharmacy. How do scientists actually take a paralytic venom and transform it into a healing medicine?
The key lies in the unparalleled precision of the conotoxins. Remember how we discussed the venom acting like millions of tiny specific keys fitting into very specific cellular locks in the nervous system?
Right.
Because the snail has spent 55 million years refining these keys to hit exactly one specific nerve switch, like a single sodium or calcium channel, without interacting with or disrupting anything else in the body. They serve as the absolute perfect molecular templates for pharmaceuticals.
I see the value in that. Because a major problem with a lot of standard human medicines is that they are chemically messy. You might take a pill to cure a debilitating headache, but the chemical enters your bloodstream and affects your entire body, causing a terrible upset stomach or drowsiness as a side effect.
Right, collateral damage.
But you're saying the snail venom is targeted like a laser beam?
Exactly targeted. To provide a concrete example, neuropharmacologists isolated a specific conotoxin peptide from a closely related species of cone snail. They studied its structure and synthesized a revolutionary FDA-approved pain medication called ziconotide, which is distributed under the brand name Prialt.
And this isn't just a mild analgesic. This isn't a substitute for aspirin. This is intense, heavy-duty medical intervention.
It is prescribed strictly for patients suffering from severe, intractable, chronic pain, often pain associated with cancer, neurological damage, or severe spinal injuries where all other treatments have failed.
Wow. How does it work?
Ziconotide works by mimicking the cone snail's venom perfectly. It travels straight to the human spinal cord and explicitly blocks the N-type voltage-gated calcium channels. These specific channels are the microscopic tunnels that transmit the sensation of pain from the nerves up to the brain.
And it just shuts them.
Ziconotide slams those tunnels shut. And the clinical efficacy is miraculous. Ziconotide is estimated to be up to 1,000 times stronger than morphine.
1,000 times more potent than morphine. That is almost difficult to comprehend.
And here is the truly revolutionary aspect. Unlike morphine, fentanyl, and other synthetic opioids, which flood the brain's opioid receptors, causing severe side effects, immense physical tolerance, and devastating addiction, ziconotide does not interact with opioid receptors at all.
So it's not addictive.
It is entirely non-addictive. Patients do not build a physical tolerance to it requiring higher doses. It simply acts as a clean, precise blockade against the pain signal.
That discovery alone is life-changing for thousands of people suffering daily from chronic pain, and that is just one application of one peptide.
What about the diabetes breakthrough we touched on earlier regarding the nirvana cabal?
That research is currently at the bleeding edge of endocrinology. Medical researchers are intensely studying the geography cone snail's weaponized hunting insulin, Conin's G1.
Right, the super-fast insulin. Recall our discussion about how normal human insulin clumps together into tight hexamers, and the frustrating reality that it takes up to an hour for those hexamers to break apart and actually start lowering blood sugar.
The sticky Lego bricks that have to be unsnapped.
For human beings living with type 1 or advanced type 2 diabetes, having to inject insulin and then wait up to an hour for it to unclump before they can safely consume a meal or bring down dangerously high blood sugar is a massive logistical challenge and sometimes a severe medical danger.
The delay is the enemy of effective treatment.
But the snail's insulin, because it lacks that sticky peptide, is naturally synthesized as a single unattached monomer. It activates in 5 minutes.
So scientists are utilizing the snail's evolutionary blueprint. They're literally looking at the specific genetic sequence of how the snail removed the sticky edge of the insulin molecule, and they are using that knowledge to design and build entirely new, synthetic, ultra-fast-acting insulins for kids and adults living with diabetes.
It represents a paradigm shift in how we approach pharmacological design. It is a perfect, tangible example of why preserving marine biodiversity is an absolute imperative. The geography cone snail isn't merely a fascinating biological oddity. It is a vast, swimming library of undiscovered medical cures.
Which leads me to a broader ecological question. Aside from offering humanity brilliant blueprints for modern medicine, what functional role does the geography cone snail actually play in the ocean? Why does the complex coral reef ecosystem require the presence of this snail?
It occupies a highly specific and vital niche as an apex micropredator. It is instrumental in maintaining the delicate balance of the reef's food web.
Let's expand on that term, apex micropredator. It implies it is at the very top of its own miniaturized food chain.
Correct. By continuously hunting and consuming small bottom-dwelling fish, the cone snail exerts constant downward pressure on those fish populations. It ensures that no single species of blenny, goby, or fleeper fish is allowed to breed unchecked, overpopulate the reef, overconsume the available resources, and ultimately push the entire intricate ecosystem out of equilibrium.
And in a beautiful twist of ecological interconnectedness, the snail itself serves as a crucial food source for some of the reef's true majestic giants. We briefly mentioned sea turtles earlier.
Ah, yes, the hawksbill.
The critically endangered hawksbill sea turtle has an incredibly specialized digestive system. They actively hunt in the rubble for cone snails. Their stomachs and immune systems have evolved to somehow neutralize or safely process the intense conotoxins.
It's amazing.
So the snail eats the fish to balance the bottom, and the turtle eats the snail to survive. It is the complex circle of life playing out quietly every single night in the dark.
But we must acknowledge that this beautiful, finely tuned system is currently facing unprecedented environmental pressure. On a global scale, the International Union for Conservation of Nature lists the geography cone snail under the category of least concern, which technically means the species as a whole is not facing imminent global extinction tomorrow.
That's good news, but
However, on a local level, the specific coral reef habitats they entirely depend upon are in grave, immediate danger.
and the primary culprit driving that danger is the death of the coral reefs themselves.
The data is alarming. Anthropogenic climate change is steadily increasing the baseline temperature of the world's oceans. Corals are highly sensitive organisms that live in a symbiotic relationship with microscopic algae called symbiodinium.
They live right inside the coral.
Yes, they live inside their tissues and provide them with brilliant colors and essential food through photosynthesis. When the ocean water becomes too warm, the corals experience extreme physiological stress. They violently expel the algae into the water.
This phenomenon is known as coral bleaching, right?
Exactly. The reef suddenly turns a stark, ghostly white, and without the algae providing food, the coral structures eventually starve, degrade, and die.
And without the complex three-dimensional coral structure to provide shelter, the fish populations collapse or migrate. And without the fish, the predatory snails ultimately starve.
And unfortunately, the rising temperature is only half of the equation. The other devastating factor is ocean acidification.
This is a chemical process that we can actually visualize with a simple kitchen experiment. Imagine taking a piece of standard blackboard chalk and dropping it into a clear glass of white vinegar. What physically happens?
A rapid chemical reaction occurs. The acid in the vinegar reacts with the calcium in the chalk, causing it to aggressively bubble, break down, and completely dissolve.
Right. Vinegar is highly acidic, and blackboard chalk is made of calcium carbonate. Well, the structural foundation of the geography cone snail's beautiful, intricately patterned shell is also made of calcium carbonate.
Specifically, a crystalline form of calcium carbonate known as aragonite.
As human industries pump billions of tons of extra carbon dioxide into the atmosphere, a significant portion of that CO2 is absorbed directly by the surface of the ocean. When carbon dioxide mixes with salt water, it forms carbonic acid, which fundamentally alters the chemistry of the sea, making it slightly more acidic.
It is a slow, invisible, but catastrophic chemical shift.
And just like the chalk dissolving in the glass of vinegar, this increasingly acidic ocean water makes it incredibly physically demanding for the microscopic baby snails to extract the minerals they need to build their protective shells.
In extreme cases of localized acidification, the water can actually begin to pit, weaken, and dissolve the existing shells of the adult snails, leaving them entirely vulnerable to predators.
It serves as a stark, urgent reminder of how deeply interconnected the entire biosphere truly is. When we allow a coral reef to bleach and die, or when we alter the pH of the ocean, the consequences extend far beyond losing a visually beautiful place to go snorkeling on vacation. We don't just lose an aesthetically pleasing map-patterned snail. We are actively losing the medical cures of tomorrow. We are erasing the genetic blueprints for non-addictive pain medicines, advanced diabetes treatments, and potentially dozens of other pharmacological miracles that we haven't even discovered yet.
It's so true.
Let's bring all of these incredible threads together for a grand recap of our exploration today. We started deep in the dark of the Indo-Pacific, watching a slow-moving, visually impaired, invisible ninja release an undetectable chemical sleeping potion over an unsuspecting fish.
We zoomed in on an atomic level to examine weaponized insulin crashing a nervous system and watched a microscopic, hollow harpoon fire at the staggering speed of a bullet.
We traveled backward through time, witnessing 55 million years of a relentless evolutionary arms race on a biological treadmill. And we concluded our journey in a modern hospital ward where the potent venom of a sea snail is actively curing human diseases and holding the incredible promise for the future of synthetic medicine.
The story of the geography cone snail perfectly encapsulates the profound wonder of marine biology. It demonstrates that the natural world is endlessly inventive, constantly surprising, and that no matter how much we think we understand, there is always an infinite amount more to learn and uncover.
We want to sincerely thank you for being here, for bringing your curiosity, and for exploring the deep, dark, and wonderfully bizarre corners of the ocean with us today. It is always a genuine privilege to share these incredible stories with you.
Before we conclude our time together, I want to leave you with a provocative question to ponder.
Let's hear it.
If a simple, brainless invertebrate moving at a literal snail's pace across a dark reef can chemically engineer a weapon so profoundly advanced that it currently holds the cure for intractable human pain, what other unimaginable medical miracles, what other astonishing biological superpowers are hiding right now in the hundreds of thousands of bizarre, luminescent, and microscopic ocean creatures that humanity hasn't even discovered yet?
Just imagine what secrets are waiting out there in the dark. Keep your curiosity wild, keep advocating for the protection of our waters, and never, ever stop wondering about the magic hiding just beneath the waves.